The amazing world of RNA contains another non-coding RNA, which is the long non-coding RNA. And this session is going to basically focus on the different aspects in which the long non-coding RNA have been found to have a role. Although there are still many long non-coding RNA whose functions are not yet known, but what is definitely very clear is that most of these long non-coding RNA have a role to play in regulation, especially with respect to gene expression. So let us look at the learning outcomes of the session. So the long non-coding RNA are considered to be greater than the 200 base pair length, and it has a wide repertoire of biological functions. Their localization, so where is it present? Is it present in the perinuclear region, or whether it is present in the nucleoplasm? Depending on that and its interactions with the DNA, RNA, and proteins, these long non-coding RNA are able to carry out varied functions such as modulating chromatin functions, stabilizing and translation of mRNA, involvement in signaling pathways, etc. Their biogenesis is slightly different from that of mRNA, and most of their diverse functions eventually focus on regulating gene expression. So you can already see if it is modulating chromatin function, then it definitely regulates gene expression. That way, most of the functions ultimately are towards regulating gene expression. Their tissue-specific and condition-specific expressions make them potential biomarkers and also therefore therapeutic agents. Now to look at the different or varied types of long non-coding RNA, majorly they are classified into linear and circular, and within the circular there are three types, that is circular RNA, ccRNA and the ciRNA. Linear has about 10 different uh, RNA that have been identified, and there could be more coming up in the future. So you have basically alliance cRNA, you have 3' overhanging one, you have antisense RNA, you have processed transcripts, you have non-coding RNA. Now you have sense overlapping, you have the telomeric RNA, you have the bidirectional promoter based RNA, you have macro non-coding RNA, and then you have sense intronic RNA. To concentrate on all of this is going to be one major task. So here this is just basically showing you that you can have various different long non-coding RNA and the sources could be varied. Now going to the biogenesis of the long non-coding RNA, when you compare it, and we're comparing it with mRNA because both the mRNA and the long non-coding RNA are transcribed by RNA pol 2. And there are just a few long non-coding RNA that have been observed to be transcribed by RNA pol 3. So when you look at how transcription takes place, again, you would find that the long non-coding RNA will also have intervening sequences. So ideally, even the transcript of the long non-coding RNA is a primary transcript, and that primary transcript should get processed. What is interesting to note at the expression level is that the long non-coding RNA is expressed at a much lower amount compared to the mRNA. And what has been observed is that there is an inefficient processing that happens with respect to long non-coding RNA. And therefore, what is observed is that if the long non-coding RNA has been processed properly, then it is exported out of the nucleus. But if it has not been processed properly or not been allowed to be processed properly, then they are generally retained in the nucleus and they would have some role to play. Now, what are the conditions thereby in lieu of which you have the long non-coding RNA being retained in the nucleus? So one of the mechanisms is that when you have the long non-coding RNA being transcribed, but when it is transcribed, it is being transcribed by a dysregulated pol2. That is because if the pol 2 is phosphorylated, then the phosphorylated pol 2 is kind of carrying out the transcription, but it is a dysregulated transcription, and so it has been found that the long non-coding RNA is then attached to by the exosomes, and once it is attached to the by exosomes, it can be degraded. So uh, this is when you have the long non-coding RNA still associated with the gene itself. Then what can also happen is that the long non-coding RNA can tether itself on the chromatin. So as the RNA pol 2 synthesizes the long non-coding RNA, and the non-coding RNA forms a secondary hairpin loop structure, here you can see that that motif is a motif that can bind to U1 SNRP or U1 snRNA, and because of the binding of the U1 SNRP, it will bind to the chromatin, and since it binds to the chromatin, the non-coding RNA now cannot escape from the nucleus. Then, of course, what has been observed is processing is not proper because the introns may have weak splicing signal. So you can see over here how the polypyrimidine tract is a little farther from the 3' splice site. Generally, it is almost touching the 3' splice site. So therefore, processing of such an intron becomes a little difficult, and hence, since it is not processed properly, it is again retained inside the nucleus. Then, of course, you can have various cis motifs and trans factors that can actually come and bind to the introns of the long non-coding RNA transcript and prevent the spliceosome machinery to carry out splicing. So again, you would find that the long non-coding RNA is basically retained inside the nucleus. Now, the processed RNA is definitely exported out, and the exporting is done by RNA nuclear export factor 1, and the functional long non-coding RNA that is there, as you can see, has a cap, that is the 5' cap. It has the introns spliced out, and it also has a poly A tail, very similar to that of the mRNA. So the mRNA definitely undergoes translation, but the long non-coding RNA have been found to bind to either RNA binding proteins in the cytosol, or they can bind to the ribosomes, as and their 5' end they have what is called as a pseudo 5' UTR. So that can enable the ribosome to come and bind. But what happens to this long non-coding RNA is still not very clear. There are some reports suggesting that it may get degraded, or it may have a role to play in the translation mechanism per se. What is also observed is that the long non-coding RNA can also locate itself or get translocated into the mitochondria, where it is stabilized by certain proteins, and then within the mitochondria it can play a role in gene expression. Then, other than mitochondria, it has been found to be associated with several other organelles as well. Like, for example, in case of exosomes, you can find that how the long non-coding RNA can bind to the RNA binding protein and then translocate it to a particular organelle. So these are different ways by which the long non-coding RNA are associated with other proteins, other complexes in the cytosol. Now, what is alliance cRNA? We'll just take a few long non-coding RNA and see from where it arises. So the LINC RNA arises from intergenic regions. So suppose you have the DNA, you have a gene here and you have another gene here, and in between the two genes there is an intergenic region which does not really have a gene, and if that area gets transcribed into an RNA, then that is what is called as alliance cRNA. You have eRNA. Now, this arises from enhancer regions. Enhancer regions are cis elements present in the eukaryotic DNA, and these enhancer cis acting elements play a very important role in increasing the rate of transcription, or not just increasing the rate of transcription, but actually allowing the transcription initiation complex to be formed to initiate transcription. So they are pro-initiation of transcription, and hence these cis-acting enhancers, these DNA elements themselves can be transcribed to give rise to enhancer RNA. Then you can have what is called as promoter upstream transcripts, and interestingly these are transcribed in the from the promoter region itself. That is interesting again. You can have antisense long non-coding RNA. So this is nothing but antisense RNA transcripts. So suppose you have the double-stranded DNA which opens up when transcription has to take place. We all know that RNA pol 2 will select the coding RNA, coding strand of RNA, strand of DNA to synthesize the transcript, which is the sense RNA. Okay, but suppose the other strand of DNA, which is a non-coding strand of DNA, gives rise to a transcript, then this transcript is what is called as the antisense RNA, and so therefore this becomes an antisense long non-coding RNA. Likewise, you have many other non-coding RNA which arise from different situations or different parts of the genome. Now moving forward to looking at the kind of its main role, that is its role is in gene regulation. So we'll look at different aspects which will ultimately come to the same thing, that it has a major role to play in gene regulation. Of course, with the thought that its major role could be in chromatin regulation, and the general idea that the RNA molecule is negatively charged because of its sugar phosphate backbone. It can interact with the positively charged histone tails, and when it interacts with the histone tails, neutralizes the histone trails, then chromatin decompaction has been observed, and change in the chromatin wound around, or the DNA wound around the chromatin. So it becomes, the DNA becomes less condensed if it is decompaction, then it can definitely influence gene expression. So we look at various different ways in which the long non-coding RNA help in chromatin regulation. So one way is recruitment of chromatin modifiers. So you have what is called as a hot tip gene, which is actually transcribed into a long non-coding RNA, and this long non-coding RNA associates itself with proteins that can actually create chromatin folds to decide a particular domain for gene expression, and then it recruits histone modifiers, and when they recruit histone modification and histones are modified, then it will affect the HOX A5 genes to be transcribed. So it influences transcription by actually recruiting chromatin modifiers, and in this case it is very clearly seen that it is recruiting histone modification. Then you can of course have, say for example, you have the long non-coding press1, okay, and this is able to bind to SIRT6, which is a chromatin modifier. Now when the long non-coding RNA is bound to the chromatin modifier, the chromatin modifier is not able to interact with the chromatin, and thereby it is not affecting the already modified histone, and so because of that the genes, the pluripotency genes, are continuously expressed. But when you have the long non-coding RNA not being present, then the same chromatin modifier will go and bind, change the modification, and by changing the modification it can inhibit the transcription of genes from these pluripotency genes. So therefore you can see how it can switch, how by not binding. So by not binding of the chromatin modifier, you can have the genes getting expressed, and by binding of the modifier, you can see the genes get inhibited, not expressed. And that is thanks to the long non-coding RNA binding to the chromatin modifier or not binding to the chromatin modifier. Then of course what is observed is that you can have the long non-coding RNA directly interacting with the chromatin, and when it directly interacts with, say for example, when you have the RNA pol 2 carrying out transcription, and if the RNA formed from that itself is looping back again in that open DNA, then that is what is called as R loop. So you have the R loop binding to several different molecules that may carry out DNA methylation. So when they carry out DNA methylation, then you have the expression of this gene happening, and that is thanks to this long non-coding RNA interaction with the chromatin itself. You can also have the long non-coding RNA interacting with an RNA DNA complex. So here it is basically acting as a trans system, and so it can interact with that, and it can, as you see over here, begin transcription of genes by RNA pol 2. So when you have the long non-coding RNA interacting with the RNA DNA over here, then you can see that expression has begun. So these are just different ways by which you have the long non-coding RNA interacting with chromatin, and on interacting with chromatin, or not just interacting with chromatin but regulating chromatin. And we all know that chromatin is definitely going to either affect inhibit gene expression or increase gene expression. Then we come to other ways by which you can have the long non-coding RNA acting, and this is more or less transacting long non-coding RNA. So as you can see over here, you have the long non-coding RNA getting transcribed. So you have the AIRN long non-coding RNA formed, which can bind to the polycomb related complex, and this polycomb related complex is of course able to modify chromatin, and because it is able to modify chromatin, you can see that transcription is not going to happen. So here it is very clear that this is going to work as a trans molecule. So it is not cis, but it is a trans regulator. Another interesting thing, and this has been observed a lot in eukaryotic system, is that once you have the enhancer region getting transcribed into the enhancer RNA, then the enhancer RNA is able to enable cohesin to loop out the chromatin, and because it loops out the chromatin, what is observant is that the enhancer region comes close to the promoter region of a gene. So it is close to now promoter region of NRIP1, and it is also close to TF5. So this enhancer is able to activate transcription from this gene as well as activate transcription from this gene. So this is again a very interesting way by which you can have the long non-coding RNA effectively affecting transcription. This is again an example of how the enhancer RNA can loop out the DNA, and when they loop out the DNA, you can see that how the enhancer region comes close to the promoter region, and because the enhancer is bound to the chromatin remodeler ensures that the pre-initiation complex is formed properly, and it allows transcription to move ahead. Then, interestingly, another thing that has been observed is that within the long non-coding RNA genes, you can have the enhancer elements present. So, therefore, these enhancer elements themselves are able to modify the chromatin to ensure that activation of a gene is happening. Now, you may also have that one long non-coding RNA can actually regulate several different elements, which in turn can regulate a expression of a particular gene. So this is also something that is interesting. So just to go by one after the other, histone modification aided by long non-coding RNA can silence genes. The eRNA helps in chromatin looping, bringing promoters closer to the transcription enhancers and therefore not silencing genes but activating genes. Enhancer element within the long non-coding RNA locus is activated on forming the RNA. And of course, enhancer elements within the long non-coding RNA locus as well as chromatin looping can lead to multiple regulation. So we again look at how transacting through protein binding. Now this is not just the long non-coding RNA, but long non-coding RNA when it is bound to a protein, how the complex of the protein and the long non-coding RNA help in various functionalities. So we all know that long non-coding RNA, because of its very distinctive motifs, can bind to specific proteins. And depending on what protein it is bound to, it can have different function. Now, for example, let's say, for example, you have the PNCTR, which is a long non-coding RNA, and this long non-coding RNA can sequester um, polypyrimidine track binding protein 1. So what happens is that because of the presence of PNCTR, all the PTBP1, that is the protein that can aid in mRNA splicing, is now sequestered within the perinucleolar region. So you can see how the long non-coding RNA, which is localized in the perinucleolar region, has an affinity to bind to the polypyrimidine track binding protein 1, and because it holds on to the polypyrimidine track binding protein 1, these are not available for splicing, and hence mRNA splicing remains incomplete. So you can see how it is affecting formation of protein, or it is affecting the RNA processing. Then what you see over here is that suppose and this NORAD is in response to DNA damage. So the long non-coding RNA can bind to a protein PUM12, which on binding to mRNA is actually stabilizing the mRNA. So when you have a competition, or when you have competitive binding of PUM12 to either the long non-coding RNA or to the mRNA, one would find that you have more and more protein coming and binding to the long non-coding RNA. So therefore, the mRNA is binding to less number of PUM12, and therefore the stabilizing effect of the protein on the mRNA is less, and therefore the turnover rate of mRNA becomes higher. So you can see how there is regulation, but how is the regulation happening? By affecting the mRNA turnover. So if more and more mRNA are becoming less stable, then they get degraded, and so you will have the expression of that particular protein getting lower. It can also modulate signaling pathways. So here you have another long non-coding RNA called FAST, and because of its peculiar structure, it is able to bind to another protein called beta TRCP. Now this protein is required to degrade beta catenin, but when the beta TRCP is bound to the FAST, it is not available to degrade the BCAT. So therefore, the beta catenin will activate the WNT signal pathway, and so on activating the WNT signal pathway, pluripotency genes are going to be expressed. So this is something that is again interesting. This happens in the cytosol, but its effect ultimately is in the nucleus. So you can see how again, because of affecting a signaling pathway, you have the effect at the gene level, by lieu of which it is either expressed or it is not expressed. Another example of transacting, but this time it base pairs not with just proteins, it base pairs with target RNAs. So what you observe is the long non-coding RNA can directly interact with the mRNA, and this RNA-RNA hybrid that is formed over here is the nucleation center for a protein complex that would actually degrade the mRNA. So you can see how the long non-coding RNA can lead to degradation of mRNA and thereby underexpression of a particular protein. You can also have the long non-coding RNA attaching itself to the 5' UTR region of an mRNA, and when it binds to the UTR region, 5' UTR region, it can activate what is called as polysome formation. And we all know more number of ribosomes that bind to an RNA, more the number of proteins are going to be formed. Another interesting thing that has been observed is that a long non-coding RNA can have sequences that can specifically interact with microRNA. And we all know that microRNA are those RNA that would go and bind to a prospective or a target mRNA, and when they bind to the target mRNA gets degraded. So here what happens is because of the presence of the long non-coding RNA, there is a competition for the MIR to bind to the long non-coding RNA or to bind to the target RNA. So in presence of long non-coding RNA, you would have less number of microRNA binding to the target proteins, and so the target, not proteins, target RNA, and so the target RNAs, mRNAs do not get degraded. That is how, again, you have control of gene regulation. So in this case also, what you will observe is that the target mRNA is not being degraded, you will have more of the protein being formed. Well, one another interesting observation is that you have many long non-coding RNA associated with pathways that are directly related to certain diseases. So we all know that P53 is a suppressor element, and this P53 is induced due to DNA damage, etc. And when they are expressed, it has been observed to, say, this is an example, they have been able to express, say, in this case, the guanine, a long non-coding RNA, and this guanine long non-coding RNA is able to go and bind to BRCA1 and BARD, which in turn is going to assemble proteins for repairing through homologous recombination or non-homologous end joining. So you can see how a double strand break can be repaired thanks to the fact that the long non-coding RNA is helping a set of molecules to act as scaffolds. So this long non-coding RNA is actually acting as a scaffold for BRCA1 and BRD, and they in turn are acting as scaffolds for proteins related to the repair mechanism. So they are basically helping in repair mechanism. Interestingly, what is also observed is this garden long nuclear non-coding RNA can also be exported out, and once they're exported out, they can bind to the microRNA of the telomerase repeating telomere repeating binding factor 2, and so you can see how, because it binds, because the long non-coding RNA binds to the miRNA, you have more of this protein being formed, and this protein is required for what? Stabilize the chromosomes by forming a cap at the telomere region. So you can see if this guanine is not present, DNA repair will not happen even if P53 is induced, and you will also see that the telomeric ends are not stabilized. So in both the ways, whether there is more of DNA damage and whether you have the telomere getting less and less stable and therefore shortening, it can lead to a condition of cancer. So you can see how the long non-coding RNA is associated with a pathway that helps in repairing DNA, that helps in ensuring that the DNA or the chromosomal integrity is maintained or the genome integrity is maintained, thereby ensuring that the cell functions not abnormally but completely normally. This is another example. So you have another long non-coding RNA which can actually enhance the synthesis of pluripotent gene, which is the MYC gene. Now, generally what happens is that uh, there is a competition between these two. So both are getting expressed, but when this gets expressed more, you will have less of MYC getting expressed. So there is a control between the two. Suppose here you have some kind of mutation and this is not getting expressed, you will immediately see that you have the MYC gene getting expressed more. So therefore, in a way, what is understood is that the long non-coding RNA is forming chromatin loops to form major expression domains. So when that happens, you will see that the long non-coding RNA recruits a protein, and by recruiting the protein, it is preventing the PVT1 to get expressed, but it does definitely allow MYC to get expressed. So you can see how selective expression of a particular gene is made possible by the long non-coding RNA. So this is exactly what is happening. It is enhancing the MYC expression. Now, one phenomenal aspect of long non-coding RNA is in dosage compensation, and this has been actually studied right from 1990s, where it has been understood that X chromosome inactivation is thanks to again a long non-coding RNA. So both active X chromosome and the inactive X chromosome, they have what is called as an X inactivation center, and within the X inactivation center are several genes that are transcribed, one of which is the XICT RNA. So again, this is regulated by other genes. So you have the XT, SIC, SIX RNA, which if present you would have inactivation not being happening, but if you have excised StRNA getting synthesized, this excised RNA will go and bind to different parts of the X chromosome, and these in turn will recruit certain proteins. So as you can see, what happens is the entire X chromosome becomes inactive because of this epigenetic change. So nothing is really happening to the sequence of the DNA, but what is definitely happening is that because of the binding of the excised RNA and these different proteins, you will have this X chromosome inactive, but this will function normally. So let us make therefore the conclusions. The long non-coding RNA biogenesis is by RNA pol 2, but its expression and processing may differ from that of mRNA, due to which some long non-coding RNA are exported and some are retained within the nucleus. Most long non-coding RNA have been found to have a role in gene expression, especially by regulating the chromatin through histone modifications or through chromatin remodeling. The long non-coding RNA can also affect mRNA stability. They can affect RNA processing, and therefore it is either directly binding to the RNA or directly binding to the RNA or through an RNA protein complex that these functionalities occur. It has been found that long non-coding RNAs are involved in pathways that are directly related to diseases, and hence they could become possible targets for therapy. The world of long non-coding RNA is huge, as you can see, with diverse functions, and yet most are associated with regulation. They can be cis-acting or they can be transacting. Their prevalence is a clear indication of the reality of there actually being no junk DNA. Every part of the DNA that is transcribed seems to be giving rise to non-coding RNA, but all these non-coding RNA seem to be having a role to play. Thank you.